sırrını çözen bilim adamı. Ama kendisi hakkında yaptığım hem akademik hem de genel basın incelemelerinde son derece mütevazi olan hocamızın beynin sırrını çözen bilim insanı sıfatına mütevazi bir şekilde çözümüne katkıda bulunmak üzere yöntemler geliştirdiğini, çünkü manyetik görüntüleme tekniğinin beynin sırlarının çözümünde gerek kendi liderliğindeki araştırmalarda gerekse bu tekniğin ve donanımın başka araştırmacıların da elini güçlendirmedeki katkıları hocamın evrensel düzeyde çok yıllar etkisi devam edecek katkılarıdır. Türkiye'de de hocamızı takip ediyoruz. Koç Üniversitesi tarafından kendisine ödül verilmiş olması ülkemizin de dünyadaki bu değerlerimizi takip ettiğinin bir vefa borcudur. Bu yüzden bu kurumumuza da teşekkür etmek istiyoruz. Hocamı fazla bekletmeden heyecanla yapacağı sunuyu yapmaya davet etmek istiyorum. Katkıları için şimdiden Türk Fizyolojik Bilimler Derneği adına kendisine teşekkür ediyorum.
Çok teşekkürler Ahmet Hoca ve aynı zamanda Bayram Hoca davetiniz için. Şimdi share screen'e geçip konuşmamı açmak istiyorum. Müsaadenizle bir dakika. Ümit ederim benim ilk slaytımı görüyorsunuz şu anda ekranda. Doğru mudur?
Evet hocam, tam ekran yapma ihtimali var mı? Hem sizi hem ilk slaytınızı görüyoruz.
Tam ekran hocam. Tam ekran şu anda.
Şu anda tam ekran, yani başka...
Tamam. Tamam.
Beni de mi görüyorsunuz aynı zamanda? O enteresan. Ama şu anda tam ekran. Peki, o zaman konuşmaya geçeyim. Tekrar teşekkürler. Hem davetiniz için hem de introduction için. Daha evvel de bahsettiğim gibi maalesef konuşmayı İngilizce vereceğim. Çünkü Türkçe benim için konuşma vermek için epey uğraşmam lazım. Lise değil, hatta ilkokuldan sonra ortaokuldan başlayarak ben hep ilmi İngilizce olarak aldım. Tabii üniversiteyi Amerika'da okudum ve aynı zamanda da çok uzun senelerdir Amerika'da çalışıyorum. İlim dünyasında her zaman tabii İngilizce geçerli olduğu için İngilizce kullanıyoruz. Hatta benim laboratuvarımda çalışan epey Türk var şu anda. Onlarla ilişkim gayet enteresandır. Bir konuşmaya başladığımız zaman hadi merhaba nasılsın falan hep Türkçe gider. Ondan sonra ilme geçtiğimiz zaman yavaş yavaş konuşma İngilizceye döner ve İngilizce konuşuruz. Peki, so I will switch to English now. The title I gave you is Neuroimaging in the Human Connectome Project and the Brain Initiative. And the topic really deals with two major initiatives that has been going on in the United States, and I think that they take place in the context of the fact that today we perceive the central challenge of the 21st century as the task to understand human brain. And I quote here Eric Kandel, who received the Nobel Prize in Physiology in 2000. When he said in this book, the book called The Age of Insight, I show the front cover of the book here. He said the central challenge of science in the 21st century is to understand the human mind in biological terms. As I said, I'm sure that many of us, of course, feel the same way about tackling the problem of the brain. But it's very interesting the way he has put this in this book. He's not just saying that we have to understand the brain, but he's saying we have to understand the brain, human mind in biological terms. Essentially what he's saying is that human mind is really a biological machine, and we can understand this biological and extremely complex machine despite the fact that it is superbly complex. But there's another reason where there's such a big focus these days on the studies of the brain, namely that neuropsychiatric disorders have become the biggest burden on the society in advanced societies. This data is from the United States, showing that neuropsychiatric disorders, the burden it imposes on the society is much larger than cardiovascular and circulatory diseases, much larger than cancer. And data from, for example, I've seen from the European Union looks exactly like this as well. So to tackle this challenge, recently in the United States, there has been major initiatives. This is in addition to the significant amount of funding that the National Institute of Health in the United States invests in brain research. So, and these were targeted programs, and one of them is the Human Connectome Project, which was led by 3 universities, Washington University, Minnesota, which is my laboratory, and Oxford University. And of course, also the BRAIN initiative that came shortly after the Human Connectome Project, and both of these projects are actually still ongoing. But these initiatives were launched with the recognition that we are facing a major challenge when we are trying to study the brain. And the challenge is the brain operates over a very vast spatial scale. So, for example, we have networks in the human brain, and we have mesoscopic scale organizations. These are approximately sub-millimeter organizations where you have population of thousands of neurons that perform elementary computations are clustered together, and the picture is for orientation domains and ocular dominance columns in the visual cortex. And of course, we have individual neurons, which is a picture is shown here where we have the cell bodies. These are these bright round pictures that you are seeing in this 2-photon image from an exposed brain. And of course, they have multiple connections and dendrites and axons. And yes, we can even go further from these cell body pictures. And each of these cells, the soma, has many dendrites. And in these and dendrites, we have these numerous dendritic spines. And going further, even in the spatial scale, that is accessible only by electron microscopy, we can look at the connections at the level of individual cells and synapses. So the complexity is immense in terms of the scale which the brain covers, in terms of its organizations that enables its function. And at the same time, we have a major problem dealing with the fact that we have to work on ultimately in the human brain. And human brain means that we have to use predominantly non-invasive techniques. There are some invasive studies in the human brain, for example, during neurosurgery, but predominantly we are restricted to non-invasive techniques, which means at the present time for human brain, we are talking about magnetic resonance imaging, which is, of course, the modality that I have been working with ever since my career after my postdoctoral work. So we know today that magnetic resonance can provide a lot of information about neuroanatomy. It can provide information about neuronal activity, so-called fMRI or functional magnetic resonance imaging. We can follow functional connectivity based on so-called resting state fMRI, and structural connectivity. We can also have access to that to a certain degree using diffusion-weighted imaging for tractography. So these are actually for brain studies, for the human brain, they are extremely important information to study human brain. But there is a problem with magnetic resonance imaging techniques. And this problem was outlined when the NIH Brain Initiative was announced in this particular article that was published in Science by Tom Inzel, Story Landis, and Francis Collins. Francis Collins is the head of the NIH and head of NIMH and head of NINDS. they stated that human neuroimaging, referring to MR, magnetic resonance, captures the whole brain in action, but each 1 millimeter cube voxel includes at least 80,000 neurons and 4.5 million synapses. So, in other words, our resolution, really compared to the spatial scale in the magnetic resonance measurements, is relatively coarse. So at this point, we were tasked with this Human Connectome Project, where the task was provided and structural connections among gray matter locations in the human brain at the highest possible spatial resolution. And this project started in two phases. Phase one was development and optimization of instrumentation and techniques. So, of course, we had to go beyond what was possible with MR techniques at the time. And then we were supposed to generate a 1,200 subject database. Now, it's important I mention the fact that we had to focus on technical developments because of the fact that, as I had mentioned, in magnetic resonance imaging, we do have this problem of signal-to-noise limitation, which limits the spatial resolution we can achieve. So one of the challenges was to improve that, and this is, of course, something that my laboratory has been active in for quite a long time. Our goal was then maximize the signal-to-noise ratio in individual images. Signal-to-noise ratio is ultimately what restricts spatial resolution, and we have to also acquire data much faster. I mean, one way of increasing signal noise is to acquire a lot of data for a long period of time, which is essentially an averaging method. But in human studies, you cannot average and signal average for days, as you can do, for example, on ex vivo specimens. So we had to also acquire data much faster without compromising the signal noise ratio. In the Human Connectome Project, there are really two methodologies that MR, or magnetic resonance imaging, uses to obtain information about connectivity. One of them is the so-called functional connectivity, and you are looking at the fact that when you are doing fMRI studies, you are acquiring images one after another in a time series, and during that image acquisition, signals coming from different parts of the brain actually oscillate. They are oscillating, but this is not noise, and in fact, this is the oscillations induced by the brain. Brain oscillates quite a lot, actually, in many different frequencies, and we can capture this we can capture some of these frequencies in the magnetic resonance measurement. And as I show you here, for example, these two points, they have these oscillations, and the oscillations are somewhat correlated, as you can see. They don't appear to be random. So you can actually correlate every single point in the brain with every other point in the brain in terms of your image, and you can generate something, what you can see here, a matrix showing the correlations of spontaneous fluctuations. Yes, these are spontaneous fluctuations. Now, you can, for example, probe this matrix in many ways. You can look at a single line along this matrix of data, and what that signal line corresponds to is actually an image like this with a seed over here, which shows that this area, in terms of its spontaneous fluctuations, correlates with all of these areas in this particular fashion. So it correlates with this area in the other hemisphere, and it correlates with the frontal areas like this, and it has anti-correlation with other areas as shown over in the brain. You can look at another line, for example, in that matrix, which would be something like this, a second point where these correlations move. So these are so-called functional connectivity data, and they are based on these spontaneous correlations that take place in the brain. What we did in the Human Connectome Project, technically, in terms of technical improvement, we did in fact increase the speed at which we can sample these correlations significantly, approximately, I would say, factor of 10. So here's some data from the standard magnetic resonance fMRI acquisition looking at these correlations, just showing you some correlations in the motor cortex area and the visual areas as shown over here. In this case, the whole brain acquisition time is 5.7 seconds. So that means that in this time series, you are acquiring one whole brain image after another for a long time. The temporal resolution is 5.7 seconds. But what we did in the Human Connectome Project is develop or introduce a technique whereby we can actually sample this much, much faster. And these are data now showing this improvement. And we are sampling the data with 0.7 seconds, the whole brain imaging acquired in this case at 0.7 seconds. And what you see is that this particular rapid acquisition improves the statistical significance of the detection of these correlations as shown here. The colors are now brighter, showing higher statistical significance. And not only that, you can start seeing areas which were not visible over here. For example, in the motor cortical networks, we expect not only to engage primary motor cortices, but also supplementary motor areas as well. We know that they are there and they should be oscillating together. And we see them very clearly in this data, but not so much over here. The other technique that is a fundamental contributor to the Human Connectome Project is diffusion imaging. And I'm sure you have seen images like this, where by following the diffusion of water in the brain, we can actually get an idea of the connections or the pathways that connect different regions of the brain, generating these very colorful images showing, for example, areas like this connecting to many other areas. And just to give you some other examples, this is another data set where you see these connectivity, the fasciculated or the bundled axons in the brain, looking at, for example, the U fibers and the fibers going from motor areas down to the spine and across the two different hemispheres. And here's an expanded version of these. These are not real, microscopic images of the fibers. It is actually a depiction of the areas where such fibers exist. And so you know that the fibers are there, but it's not a microscopic like an optical image picture of those fibers. And again, the techniques that we developed in the Human Connectome Project allowed us to go to much higher resolutions. For example, at that time, the best resolutions for diffusion-weighted imaging were approximately 2 millimeter isotropic. And here, what we are doing is we are looking at, this is a probabilistic tractography. We are looking at tracks that are well known to exist on the brain, the corticothalamic, corticobulbar, corticospinal, and striatal projections. And we know that these projections in the brain should actually have a very nice organization in this particular slice indicated by these green arrows. And at the time, what was possible in terms of diffusion imaging, you can get a hint of these, the presence of these projections, but at the same time, we have a lot of other things here, which are actually, if you like, false positive activation. With the Human Connectome Project data in developments, we achieved significantly better resolutions. I mean, it doesn't sound like too much, maybe going from 2 millimeter to 1.25 millimeter, but you have to realize that signal to noise is proportional to the cube of this dimension. So we are cutting down the voxel size, the three-dimensional voxel size, by approximately 2 in this case. And in the Human Connectome data, these projections, in fact, are visualized very beautifully, demonstration of the importance of the spatial resolution in studying the brain. And today, there are many Human Connectome-like projects. Of course, this was what I was showing you is the original project that went from 2010 to 2016. We did collect 1,200 subjects. This is a database now that is heavily used for analysis. And from this Human Connectome Project, the number of publications are increasing extremely rapidly just using this database from many other labs. And but this original Human Connectome Project in fact, gave birth to many other Human Connectome Project-like projects. For example, there's this lifespan studying babies, children's and adults, connectomes related to human disease. There are many such projects running right now in the United States, mental illness, epilepsy, Alzheimer's, etc. Developing HCP, prenatal and neonatal studies based on the Human Connectome Project. The ABCD project, Adolescent Brain and Cognitive Development, a very ambitious project that is going to look at development over a very long time. So all of these are being now run by the tools that we developed at the University of Minnesota, and then are being collected at the in a data repository at Washington University. And there are now worldwide many other efforts like that as well. The Human Connectome Project. There's for example, UK Biobank, where they are planning to look at 100,000 people. And these are various initiatives at many different countries. But so I would consider a Human Connectome Project, and NIH considers it a success. But still, we have not made an incredible dent in the problem that was identified, and I mentioned at the beginning. Namely, okay, we did progress compared to what was possible technically, substantially. I would say we progressed, but we are still in the millimeter range in terms of the spatial resolution. And this causes still problems. I mean, we are looking at fMRI and this diffusion imaging data to obtain these information. But if you look at the raw data, I mean, it's one thing to look at these beautiful pictures that depict and very seductive, actually, pictures that depict information about brain. But it's another thing to look at the raw data. And the raw data is actually noisy. fMRI and DMRI data are noisy. These are low signal-noise ratio measurements. So I will demonstrate to you with showing you the raw data for functional imaging for visual stimulation. So in this case, the subject was in the magnet, and the visual stimulation came during these periods as shown over here. And each of these boxes over here, in fact, represent the time course of that the signal in that voxel during the experiment. So when we look at these data, they look already pretty noisy. I mean, there are voxels where you can identify right here that these are these changes that track the visual stimulation. That's clear. Well, sort of clear. But here's another voxel where you can actually, by eye, see the fact that in this voxel, there is a response to visual stimulation. And but what about this voxel? I mean, it is kind of questionable. There may be something there, but it's very poor signal to noise. And what about that one? I mean, again, looks like maybe there is something there, but it is in fact rather low. So I want to make the point that MR measurements are intrinsically low signal to noise, and this is what we have to fight in MR and what we have been fighting for quite a long time in my laboratory. So one of the things you can do to remedy that problem is to go to high magnetic fields, and this is the specialty of my lab. And high magnetic fields, these are much higher than what is clinically used. They have their own challenges, and they are not quite straightforward, but in principle, they do provide gains in signal to noise, especially for functional imaging. So, for example, in the Human Connectome Project, the main data acquisition was carried out at 3 Tesla, but we did run a small pilot project, which was carried out 7 Tesla, on a subset of subjects, which were also scanned at the 3 Tesla. And I'll show you the fact that this higher magnetic fields do indeed provide gains. For example, here's functional connectivity based on seeds, something I've shown you before. Here's a seed, and you are looking at correlations in the fMRI data with this particular area. So this area is correlated strongly with this area on the other hemisphere, anti-correlated with the blue areas. And, well, you can see some correlation, and this is the correlation values over here. This is 3 Tesla data. And, yeah, that's the seed over here again. And then when you go to 7 Tesla data on the same subjects, you can see that all of a sudden the correlations are much, much more clear, and they are extensive, and the correlations values themselves are even higher. Here's another example. Again, this is 3 Tesla data, and with the seed this time is in the putamen. Right over here, it's a subcortical region. And when you are looking at the 3 Tesla data, so we are asking what is the connection or correlation in the fluctuations with this particular seed in the subcortical region putamen. And when you look at the data on the cortex, you can see that it's around 0, essentially. It's just going back and forth around 0. It's very low. It's really noisy. So from this data, you really, at 3 Tesla, you don't have any information or much information about the connectivity of putamen functionally to the cortical gray matter. But this is then when we look at 7 Tesla data, and all of a sudden we indeed see these correlations very strongly. So there are pathways to go improve SNR, and one of them is high magnetic fields. And as then in the Human Connectome Project, in fact, we demonstrated this very nicely. The same is also true for diffusion imaging. SNR gains from high magnetic fields are important. Here is a tractography again. It's a tractography of tracks that are going through, they are perpendicular to and going through this particular plane. These are face, hand, and foot areas for these particular fibers that we are following in the internal capsule and the nucleus VL in the thalamus. And there should be this sort of a sonotopic organization with the face, hand, and foot area. And the 3T data, which is already very advanced compared to the regular data that is out there, you can get a hint of the presence of this sonotopic organization, but it's not quite clear. But when you look at the 7T data, you can see this organization quite nicely. These are these 2 different slices in the IC. You see it very beautifully. And in the VL, it's not as clear, but it's still showing you face, hand, and foot. And one of the most important contributions of this gain in spatial resolution that comes at the high magnetic field is the connections to the gray matter, so-called sulcal or gyral bias problem. When you look at connections in the gray matter from all of these different areas using these fiber tractography, the connections go predominantly to the gyral peak, as you can see this by these red areas. So this is not unexpected because of the fact that for these fibers to make connections, they of course have to bend and go into the sulcal walls. And if you don't have enough resolution, you may not be able to capture that bending of the fibers. But when you go to 7 Tesla with much higher resolution, you can see that you capture more of those connections in the on the cortical gray matter. But nevertheless, as you can see, we have improved over 3 Tesla, but it's not really quite perfect yet because there are a lot of areas where the connections are still missing. So some of these inherent problems that are still visible in this particular case were really tackled by the BRAIN initiative. So BRAIN initiative is different from the Connectome Project. It's an initiative that was launched approximately 10 years ago now, or slightly less than that. And there was actually a committee that was put together to define the priorities or areas that had to be focused. So, as I had mentioned to you, the fact that the brain operates over very large spatial scale was very much recognized by this committee, and their executive summary called for 6 priority areas, and number 2 was maps at multiple scales, and called for coverage to synapses to whole brain. And for human studies, however, of course, synapses to whole brain is really beyond anything we can dream of today. It was more or less reduced to, for human studies, the goal was reduced to imaging of neuronal ensembles, circuits, and larger scale networks, circuits of circuits. There was even a spatial resolution goal that was attached in this. Essentially, it called for whole brain imaging with 0.1 microliter or smaller voxel volumes. So 0.1 microliter voxel volume corresponds to approximately 0.5 millimeter isotropic dimensions. Now, idea is, of course, synapses to whole brain is fantastic. This is what we have to get to, and we can get to something like that in in animal models. This is a paper from the Genelia Farms group looking at zebrafish, actually larvae of zebrafish, so baby zebrafish if you like, and where they were able to record using 2-photon calcium imaging. This is optical imaging. Many neurons in the brain, as you can see over here. But this is wonderful. There are, of course, there are reasons why you can do this in the zebrafish. Now, this is not even adult zebrafish, but if you look at the baby zebrafish, it is first of all translucent to light. The light can go through it. In the human brain, light cannot go through the human brain. And the second thing is the number of neurons is considerably limited. So we would love to do something like this in the human brain, but how do you do that? I mean, this is really a major problem. And so what we have to do essentially in the Brain Initiative is really strive to expand this range. So this is data I already showed you from the Human Connectome in this functional connectivity. And these are these mesoscopic scale organizations in the brain. As I mentioned, these are orientation domains. They exist in approximately sub-millimeter and spatial scale. And using high magnetic fields, we can in fact, we can cover this particular scale. We can go from here to here, and this is, and then what we need to do is really push for technologies where we can push the spatial scale with magnetic resonance to even to lower and lower resolutions. Now, as I already just said, we have already demonstrated using very high magnetic fields, this mesoscopic scale can be reached in the human brain. So here are actually the first human brain images, functional images of these orientation domains. So these, all of these areas colored in different colors are neurons or population of neurons. There are about few thousand neurons that respond to different orientations in a visual presentation. And these are the so-called ocular dominance columns, which are superimposed on this in the same area. These are one eye versus other eye. The cells that respond to two different eyes are segregated in this particular fashion in the visual cortex. And today, using some such techniques, this kind of work actually has really taken off. It's a primary research area right now to look at these mesoscopic scale organizations in the human brain using magnetic resonance techniques that rely on ultra-high magnetic fields, predominantly 7 Tesla. So they are usually using resolutions like 0.8 millimeter isotropic. This is kind of the limit where the sensitivity allows us to work at 7 Tesla. There has been many successful mapping of mesoscopic functional organizations, for example, direction of motion columns and layers in the human area MT, auditory cortex columns and layers, layer-dependent activity in the primary visual area in the sensory motor cortex, columnar segregation of magno and parvocellular streams in human V2, etc. There are, and the list grows, by leaps every year today. And I can give you an example from our studies on the auditory cortex. So when we started working on ultra-high fields at 7 Tesla, we first demonstrated, for example, in the primary auditory cortex, there is a tonotopic organization. This was known from animal studies, but until the 7 Tesla study from us, this organization, tonotopic organization, was not demonstrated in the human brain. So we were able to do it and show that there is a mirror symmetric, as you can see over here. The different colors represent different frequencies of the auditory stimulus, and there is a mirror symmetric tonotopic organization. We subsequently were able to map those tonotopic organizations in even smaller structures in the auditory pathway. For example, here is the inferior colliculus. It's a very small structure, and we were able to show that there is a tonotopic organization there as well. And subsequently, in other areas in the medial geniculate body, right over here in the brain, the presence of that tonotopic organization again was mapped. So we are now talking about these mesoscopic scale organizations. And finally, we came back to the primary visual cortex, now acquire the same data with much higher resolution, so that we can actually see not only the tonotopic organization on the surface, but how that organization is affected across the cortical layers. And this is approximately 2 millimeters thick, 2 to 3 millimeters thick cortex. And you can see that there are regions where this tonotopic organization is constant across this cortex. And we were able to, in fact, show that there's a differential participation of different layers in terms of auditory processing. So-called auditory columns, that was the first paper demonstrating the auditory columns. So we can do mesoscopic scale functional imaging at 7 Tesla, but signal noise is still poor. It is a limitation. And we need to go to higher magnetic fields, and we have to develop other complementary technologies to increase the signal to noise and so-called contrast to noise. And for us in CMRR, that meant going to 10.5 Tesla. This is now the highest magnetic field available for human imaging, and of course a lot of other related technologies. So we've been doing this under the Brain Initiative support. And here's the 10.5 Tesla magnet. It's a project that started also about 10 years ago, but it is coming to fruition at the present time. It's a very large magnet, it's in terms of accomplishing something like this technologically, it is quite a daunting task, but it's possible. You can see the dimensions of the magnet by comparing it to the human over here. And but nevertheless, when you go to 10.5 Tesla, we were able to show that you can get significant signal to noise gains relative to 7 Tesla. For example, here, these are calculations, but experimentally verified, so-called calculations, because they are calculating the ultimate gains that we can achieve. So theoretically maximum gains. In the center of the brain, we can get as much as a factor of 2.5 gains, going to 10.5 Tesla and 7 Tesla compared to 7 Tesla. But in the surface, we gain less. But there are other technologies which are devoted to improving the surface. Even though in the surface, from going to 10.5 Tesla, we gain only a factor of 1.5. By using these other synergistic technologies, which I will show you in a minute, we can actually gain as much as factor of 7 or 8 on the surface. And the technology that we have to rely on is using these high-density detectors. So each of these loops are a simple detector. Typically, these days, on a clinical machine, you may have RF coils which have 16 such loops or 20 such loops, maybe on very advanced machines, 32 such loops. But this coil is 128 such loops. It's an engineering challenge. You can see some of the loops here and the little electronics that are incorporated with them. It's an engineering challenge, but a challenge that is realizable, and we are a long way on that path. The other important development to gain signal to noise is image reconstruction. So there are techniques now based on denoising, so we can actually, without touching the signals that we want to see, we can suppress noise quite a bit. And deep learning is also coming into play quite a bit these days for image reconstruction for improved signal to noise. We just published a paper, it appeared in Nature Communications, based on these denoising techniques, and I want to show you what is possible with them. So here you are seeing a functional image. It's a visual stimulation in the human brain. We are looking at the visual cortex, and we have acquired data in blocks. So this is one run, and 3 of those runs, and 5 of those runs. So it gets longer and longer data acquisition. You have to have a very good subject who actually is willing to tolerate such long acquisitions. And you can see that functional image with a single run is not very good. You don't detect all the areas, but as you go to longer and longer runs, of course, you are improving the signal to noise because you're acquiring many more data, and you see that a functional image that tends to plateau starts appearing at 3 runs and 5 runs. But this denoising technique that we introduced, we call it Nordic, and then if you now use that technique, and on a single run. So this is a single run processed with this denoising technique. And you can see in a single run, we get a functional image somewhere between 3 and equivalent to somewhere between 3 and 5 runs approximately. And it's really a major jump. I'm very excited about this particular development. So we're getting approximately a factor of 2 or so improvement, if you like, in the signal to noise. And for example, if you go to very high resolution imaging, in this case 0.5 millimeter isotropic, which is actually 0.1 microliter. This is the targeted goal in the BRAIN initiative. When you acquire the data and then you acquire it with process it with Nordic, these are some images that you can see. It takes 10 averages of those images without I'm not sure why.
Hocam mikrofonunuzu bir daha açabilir misiniz? Açık bir parazit mikrofon vardı, kapattım.
It was not from you.
Açtım tekrar. Duyuyor musunuz beni tekrar?
Evet hocam, gayet net. Teşekkürler. Devam edebilirsiniz.
Evet, burada so you see here 10 averages of the normal image. It's approximately you would get with Nordic processing only of 1 acquisition. So looking at functional images, these are actually signal changes we are expecting to see during activation. Under the standard condition, you really don't see anything, and you can see it very nicely with this Nordic processing. And what you can, in fact, obtain these beautiful functional images in the visual cortex at this extremely high resolution. So with this technique at 7 Tesla, we have already hit the BRAIN initiative goal, but we have a long way to go, and we can combine this technique with 10.5 Tesla, and these are the very first 10.5 Tesla functional images, and now we have even gone beyond the resolution goals of the BRAIN initiative with 10.5 Tesla, and we are aiming, we anticipate that we will be able to reach approximately 0.01 microliter voxel volume as shown over here, combining these kinds of techniques, the kind of engineering development that I showed you with respect to detectors and the 10.5 Tesla. So I started by showing you that in the Brain Initiative, there was a challenge about the fact that 1 millimeter voxel includes at least 80,000 neurons. And that was a challenge that was put down to the scientific community, to the imaging community. And at the present time, approximately, less than a decade after we started this project, we are talking about not voxels which contain 80,000 neurons, but approximately 800 neurons. So we've come a long way, and there's still some development to do. And I think the next decade of neuroimaging will be able to achieve a very different, substantially different capability in terms of studying the human brain. With that slide, I'd like to end my talk. And of course, there are an incredible number of people who have contributed to this work. And this is a picture of the Center for Magnetic Resonance Research that I lead at the University of Minnesota. And there are quite a lot of people with many different contributions that have to come together to be able to do these studies. For example, very large engineering group as shown here. Without them, it would not be possible to develop these techniques. And data processing group, Steen and Ahmet, major contributions in terms of improving signal to noise. Of course, I talk quite a bit about the Human Connectome Project, and that's a project which is a consortium. We were responsible for developing the techniques for image acquisition and reconstruction, but University of Oxford, for example, developed a lot of techniques for image analysis, so did Washington University. And so it's really a three-way collaboration between these three institutions. And in particular, I'd like to acknowledge my co-principal investigator, David Van Essen from WashU, for the Human Connectome Project. With that, I end my talk, and I thank you for your attention.
Well, thank you for this excellent talk. Şimdi bu konuşma tartışmaya açılmıştır. Saygıdeğer hocalarım, kıymetli katılımcılar. Kamil hocama sorularımız varsa sorularımızı alabiliriz.
Bir şey sorabilir miyim? İzninizle Kamil hocam, ben bir şey sorabilir miyim?
Tabii, buyurun.
Hocam, bu işitme yolaklarında inferior colliculus'ta, geniculat nucleus'ta farklı renklerin oluşması acaba perception'ın sadece kortekste değil de bu işitme yolaklarında başladığının delili olarak sayılabilir mi?
Evet, sayılabilir. Yani ben size sırf tomotopiyi gösterdim, fakat tomotopinin dışında da bazı şeyleri belirttik. Bu inferior collicus mesela bandwidth of hearing, etc. So dediğiniz gibi bu merkezlerde already there is actually quite a bit of processing going on before the cortex.
Peki bununla bağlantılı olarak aynı frekansa sahip farklı kalitede seslerin quality is different işitme korteksinin tabakalarında aynı rengi oluşturması farklı kalitedeki seslerin, aynı frekansa sahip seslerin nasıl algılandığını açıklayan bir delil olarak kabul edebilir miyiz hocam?
Yani bugün okay, so sorry, I'll speak, switch readings again. So based on the experiments we have, we are unable to say that. Of course, it's possible. There is actually, as you mentioned, there is quite a bit more processing that goes on in the auditory perception besides tonotopy or bandwidth of the sounds that are being perceived. But so far, at least to my knowledge, nobody has really looked at these higher order processing in these other areas like the inferior calculus.
Teşekkür ederim.
I'm guessing that it's possible that there's some of that processing goes on in these structures as well.
Teşekkür ederim hocam.
Evet, başka sorumuz var mı? Sorusu olan lütfen direkt sorsun çünkü ekranda herkesi göremiyorum.
Ben sorabilir miyim Numan Ermutlu?
Lütfen Numan Hocam.
Çok teşekkürler sunumunuz için. Ben şeyi merak ettim. Bu olaylar içinde bu konnektomla ve bu bağlantılarla ilgili bu EEG'nin, Event Related Potential'ların geleceğini nasıl görüyorsunuz? Bunlara entegre olabilecek mi yoksa bizim dükkanı kapatmamız mı lazım?
Dükkanı kapatmayın. Entegre olacak tabii ve hatta Human Connectome Project'te aynı zamanda bahsetmedim bu konudan ama sırf MR'ın yanında, magnetic rezonansın yanında MEG, Magnetic Encephalography da yaptık. 1200 kişide değil ama sadece 50 kişide. Bu datada bizim, this data is also available publicly and the people do use it. It has different information. So I think EEG and MEG is really quite complementary, and there is still information to extract from about so-called functional connectivity, which is complementary to MR data. So I think it's too early to dükkanı kapatmak için beklememiz lazım biraz daha. Ve hatta kapatmamıza hiç lüzum yok.
Çok teşekkür ederim. Sağ olun.
Hocam bu konuyla kısmen alakalı, kısmen de daha geniş bağlamda ben bir soru sormak istiyorum.
Buyurun.
Sistemler fizyolojisini bir süredir askıya aldık veya popülaritesini kaybetti. Gittikçe molekülerleştik, genetik tabana doğru indik. Acaba burada gelecekte beynin anlaşılması dahil veya keşfedilen moleküler bilgilerin toplam insan vücuduna aktarılması konusunda fonksiyonel MR ve bu görüntülemelerin katkısı ne olacaktır? Yoksa bu moleküllere gitme süreci de yani genom projesi hakkında daha doğrusu düşüncelerinizi öğrenmek isterim hocam.
Very good question, and but I would say that of course I talked about the fact that we have to cover a huge scale of organization, and from the imaging point of view, I sort of stopped at approximately the connectivity at the electron microscope level, but another order of complexity comes in because of all the molecular processes that go on that support the brain function. So we have to cover that too. So in principle, we have to have information about everything, if you like. Whether at what point we can bridge that information so that we can have a uniform or unified understanding of brain activity, that will come, but in the not quite at the present time. Although there are hints where we are bridging those things, and I will mention that to you. So we have to study the molecular processes, the genetic information, and as well as the system level processing. And one of the things that is emerging from the Human Connectome Project, for example, is that there are studies looking at expression of genes in the brain and correlating it with particular functional connectivity patterns that we see using these system-level methods like MR imaging. And there have been papers published on this already. And interestingly, of course, canonical genes that are sort of expressed in many areas, they seem to be constant in their expression in many regions of the brain. They correlate with these functional connectivity patterns. And are we surprised that there's some sort of a relationship between these system-level measurements and genes? Of course not. They are linked at some point. But establishing that link is difficult. It's a big step, but there are already actually efforts that try to bridge that gap, and I think this will increase as we go on with time, and that we will have ultimately, we really have to go from molecules and genes and gene expression all the way to system level networks as accessed by MR.
Hocam bir soru da ben sorabilir miyim müsaade edersen? Bu görüntülemede özellikle konektiviteye bakarken daha yüksek zihinsel işlevleri değerlendirirken time constant'ı daha büyük olan işlevler ne şekilde görüntüleniyor? Onlarla ilgili gelecekte nasıl bir takım gelişmeler olabilir? Halihazırda daha hani kısa zaman dilimleri içinde gerçekleşen olaylara dönük görüntülemeler var ama daha geniş executive function veya cognitive functions'la ilgili frontal lob fonksiyonlarıyla ilgili neler görüyorsunuz? Sizin bu konudaki beklentiniz nedir?
So I of course showed you predominantly studies in the visual cortex, and part of the reason we work on the visual cortex quite a bit is, of course, visual cortex is maybe the best understood area of the brain. There's quite a lot of information on the visual cortex. And one of the things that we focus on predominantly is the development of techniques, tools. In fact, Brain Initiative itself focuses on tool development, emphasizes tool development. And when you are developing tools, you don't necessarily start with dealing with complex processes in the brain. You stick predominantly to areas that are understood as best as possible. So in my lab, as a result, auditory cortex studies or visual cortex studies dominate because we already know quite a bit. And then when we improve the tools, we can actually see if we are improving the information content relative to what is known from electrophysiology or optical imaging. Now, I explain this to you because my bias to showing visual cortex does not mean that there is a lack of effort to study other areas or higher cognitive functions, in the frontal cortex, etc. This goes on quite a lot. And with executive functions, there's a lot of work coming out of even Human Connectome Project about executive functions and the frontal areas, or even using what happens to these functions under neuropsychiatric diseases. When you ask me to give a lecture, however, I usually don't talk about those things because I'm focusing on development or the improvement of the techniques, and then I tend to use visual and auditory. But there are many other laboratories that use either the data that we generate or the tools we generate, use those to study higher order functions. And I think this will improve with time.
Yeah, thank you very much.
Any further question? Kamil Hocam, sanıyorum yok. Gerçekten bu mükemmel konuşmanız ve dahası insanların da kullanarak insan beynini daha iyi anlamasına yönelik geliştirdiğiniz teknikler için sizlere bir kez daha Türk Fizyolojik Bilimler Derneği adına teşekkür ediyoruz. Başkanımızın da dediği gibi yüz yüze yapacağımız bir toplantıda sizlerden, ilminizden faydalanmak üzere Türkiye'de sizleri misafir etmek istiyoruz hocam.
Teşekkür ederim. Ümit ederim öyle bir şey yakında gerçekleştiririz.
Peki Sayın Başkanım, Bayram Hocam, ben sözü size aktarıyorum. Tekrar herkese iyi akşamlar diliyorum.
Ahmet Hocam teşekkür ederiz. Kamil Hocam ağzınıza sağlık. Çok teşekkür ederiz. Katıldığınız ve bu engin bilgilerinizi bizlerle birinci elden paylaştığınız için temennilerinize katılıyoruz. İnşallah yakın bir zamanda pandemi biter ve sizleri memleketinizde, ülkenizde misafir etme onurunu bize yaşatırsınız. Değerli arkadaşlar, Türk Fizyolojik Bilimler Derneği'nin 46. Ulusal Fizyoloji Kongresi ikinci gününü de bu şekilde tamamlamıştır. Yarın sabah Türkiye saatiyle 10.30'da sözlü bildirilerle 3 salonda oturumlarımıza başlamak üzere. Şimdilik hepinize iyi akşamlar diliyoruz. Hepiniz hoşça kalın, sağlıkla kalın. Kamil Hocam selamlar, saygılar.
Çok teşekkürler. Benden de selamlar, saygılar hepinize.
İyi akşamlar.
Hoşçakalın.